Hello, everyone. Thank you for joining us today to talk about microbial strain typing for real-time quality control and source tracking using FT-IR spectroscopy. And this is a collaborative presentation between Bruker and Certified Group today. And we will be going over how to use some of the instrumentation from Bruker to accomplish microbial strain typing, which we do within our group here at Certified Group. So just a little bit of background about me to get started. My name is Alex Brandt. I'm the Chief Science Officer with Food Safety Net Services, which is a company within the Certified Group company. And really, I do a lot of functions for the company here, but I started back in 2014. And prior to that, I was a graduate student that did a lot of research with food production plant environmental sampling and pathogen subtyping. So a lot of my focus on research was on looking at microorganisms in the environment of the food production plant and trying to decipher the linkages between different isolates that we had found over time. And so when I joined FSNS and Certified Group in 2014, I was brought on to provide a lot of technical troubleshooting to customers with regard to their methods, their results, and also their production plant issues where they had contamination problems. So I still do a lot of that work now, going out to customers to help them out with issues. And so I can see the value of microbial subtyping in a lot of that work. So that's really what I want to focus on with you all today and show you how some instrumentation can possibly help you with that, that instrumentation being provided by Bruker. So in addition to the work that I do with customers, I also oversee our Lab Plus department that does contract research work and specialized testing. And a significant portion of this specialized testing that we do is really focused on microbial subtyping in order to use the data that we can get from that for source tracking and environmental mapping. FSNS also has a software program called EMMA, which is our environmental monitoring software, which also helps with source tracking. So all of this can kind of go together as one big solution to help you with finding and troubleshooting contamination events in your processing plant. So let's start off kind of with some basics here. So microorganisms in food are actually very complex groups. Food products can have very diverse and complex microbial populations. And these are all influenced by various things, including the primary production environment. So think of things like fields, farms, and feedlots, the processing facility environment, their packaging systems, the distribution chain that those foods go through, as well as what happens to them when they're handled by consumers. So these microorganisms that we can find in food can include good organisms, such as probiotics, bad organisms, such as pathogens, and the ones that are just plain ugly and nasty that we call the spoilage organisms. Hence, we have the little graphic here from our good friend Clint Eastwood's movie, The Good, Bad, and the Ugly, which kind of describes those different groups that we have in food products. So you may ask, why would I want to track a microorganism in a food product and throughout its production chain? Well, there are many reasons to want to do that. Maybe it's a pathogen, and maybe we need to decipher where it's coming from, such as a niche site in the facility or a continuous entry in raw material where that pathogen is continually being introduced into the production process. Maybe it's a spoilage organism, and it's spoiling products before their normal shelf life, and we really need to determine how it's getting onto the products. So it could be some dirty equipment, contaminated raw materials, or various other things that are causing this spoilage organism to find its way onto our products. Or maybe it's a probiotic. Maybe it's actually a good organism, and we want to determine if our starter culture that we're putting into it is present in the finished product after packaging and going through the entire process. So there's various reasons that we might want to be able to track these strains over time and see where they're at in the process and be able to link them together. So how do we do this? Well, there are various subtyping methods or methods where we are able to look really deeply at the strain of the organism. And there are a lot of different technologies that are out there? Well, there's a lot of technologies that exist to provide the species identification of microorganisms from food. However, there are many cases where we need to classify organisms beyond the species level. So we need to know if two isolates of the same species are the same or if they're different. So that's where microbial subtyping technologies come in, and they allow us to do this differentiation between isolates. Historically, a lot of these subtyping technologies have been DNA-based, such as pulse field gel electrophoresis, which a lot of people are familiar with, or also known as PFGE. A lot of people recognize that acronym. Riboprinter, multi-locus sequence typing, or MLST. People recognize that acronym quite a bit as well. And whole genome sequencing, or WGS, another acronym for you. WGS really, in the present day, is the gold standard, but that takes several days and it's quite expensive. So there also are some regulatory issues with WGS and using it for subtyping. Well, one thing that's really a big con to a lot of people using it is that you will end up knowing the species of the isolate and knowing all of its genetic makeup. So that then allows it to be tied to regulatory agency databases such as FDA, USDA, and CDC that utilize databases such as Genome Tracker. And that has other metadata. So if that is out there, then you could potentially be linked to a foodborne illness outbreak. Knowing the species is also an issue if you identify a pathogen. So your food products may not be on hold, they may be in commerce, and if you identify a pathogen through your subtyping method, that could lead to a recall. In addition, knowing all of its genetic makeup is also an issue because it can be tied to other isolates, and you know all of the virulence factors and antibiotic resistance profiles. And then if you can, as I mentioned, get tied to a regulatory agency database with metadata in a clinical case, you may be looked at as the source of a foodborne illness outbreak. So a lot of food industry entities really avoid using whole genome sequencing and rely on other methods to do their subtyping, even though it's the gold standard and even though it gives you the most information about linking isolates together. Other subtyping methods that are available that are not whole genome sequencing, such as PFGE, Riboprinter, and MLST, however, also require you to know the species, or even by conducting them, you will find out the species in the process of analysis. They don't provide as much information as whole genome sequencing, but you still get that species information, which is problematic in some cases. Also, some of these technologies are limited to only certain species. For example, the CDC PFGE protocols only cover about 10 species of foodborne pathogens. So what about all the rest of the foodborne pathogens? What about the spoilage organisms? Can't really use PFGE for all of those. And many of these technologies can be a multi-day process in some cases. So when a few years ago, when we were looking at new subtyping technologies to bring on to be able to track these organisms through people's food processes, we identified several needs. And we identified that the needs that we had were for a system that could, one, subtype all microorganisms, not just select species. Two, perform the subtyping without the need to identify the species. Three, perform the subtyping without determining the species in the process. Four, perform that subtyping quickly without the need for days of analysis time. And five, perform the subtyping relatively inexpensively so more isolates could be run. So we did some investigations, and behold, we found the Bruker IR biotyper. So Bruker created this technology called the IR biotyper specifically for microbial typing. And this is really meant to help with real-time quality control and source tracking, so that way you're not taking days to get information, but only a few hours. So when we looked at this technology, it was very promising, and we have onboarded it here at Certified Group, and it has been a very, very useful tool for us. So how does it work? Well, it generates a fingerprint from a microorganism that allows it to be compared to others. It's not a genetic fingerprint. It uses FT-IR spectroscopy to actually measure vibrations of carbohydrate constituents that are present and many cell surface molecules such as glycoproteins and lipopolysaccharides. So again, it is not DNA based, it's not a genetic fingerprint, it is actually looking at cell surface structures and molecules and the profile of those that are present in different strains. So many people may actually be familiar with FT-IR and the principle of using it for material identification, such as foreign materials. We actually use infrared spectroscopy quite a bit for foreign material identification. But this particular application is actually set up in a high throughput 96-well format with software that's specifically set up to do microbial comparisons. So this is a very important principle because it allows people to actually compare organisms without having to generate data that would be tied to PFGE or whole genome sequencing data that the CDC or FDA or USDA would have. They don't use this technology for any of their source tracking and microbial subtyping that they do, which was a very ideal thing for us when we looked at this technology. Also, the Bruker IR biotyper is not tied to any sort of biochemical identification information. It will not give you a species identification. It's not a biochemical identification method replacement. So think of methods out there like the Biomirror Vitec, which gives you a biochemical identification. This technology does not do the same thing, and it will not give you an identification of a species, which is actually good in some cases. For instance, in the Listeria monocytogenes use case, let's look at that. In certain types of food products, it makes sense to only look for Listeria-like organisms rather than looking for Listeria of a particular species, especially monocytogenes, which is a pathogen. So this technology allows you to make comparisons for these Listeria-like organisms without identifying the Listeria species. And this is very important for regulatory aspects in reacting to Listeria species versus Listeria monocytogenes positives, because if you get a non-pathogenic Listeria species, so anything other than Listeria monocytogenes, it will not necessarily implicate a ready-to-eat product if you actually find a positive. But if you have to identify something to the Listeria monocytogenes level, it may cause product to be implicated and require some sort of a rework. So this will not give you that species information for monocytogenes, which is very nice. So we can actually function on just comparing things as quote-unquote Listeria-like organisms without getting that identification information, but still allowing you to track those throughout the process. That was one of the key functions of this technology that we really liked. The utility also comes from being able to test multiple isolates to make comparisons. It's a comparison tool. So if you just test one isolate, you're not going to accomplish too much. So you need to actually have multiple isolates and even possibly a bank of isolates, quite a few, to be able to get good information out of this. So how does it work? It's actually pretty simple. The first step is to obtain a pure culture of a microorganism. We cannot run this off of enrichments or off of mixed cultures. So you have to actually get an isolated organism on a agar plate. The only limitation to this is that you must be able to take that and make a homogeneous suspension. So any bacterial organism, really, that can grow on non-selective media such as tryptic soy agar, blood agar, etc., is definitely possible because we can make a nice homogeneous suspension of that. We can also do others, but we would have to validate a different growth procedure, such as lactic acid bacteria and MRS agar. Still can be done, but we have to validate a different procedure. We can also use it for yeasts. It's not very optimal for molds because it's difficult to make a homogeneous suspension of them between the spores and the hyphae, so we tend to try to use this on things that are pretty basic organisms, like we said, like yeasts and bacteria, that we can homogenize very well. Once we have that pure culture, then we need to streak a plate to make a lawn so that we have plenty of growth to work with. So the key to success with this is ensuring that your growth protocol for making this lawn plate is very strictly followed, so that results are repeatable from one run to the next. If we vary temperatures or media, that can cause things to go out of whack with regard to the cell structures on these organisms. So a consistent incubation temperature and time really is critical so that the cell surface structure is uniform from one run to the next. So once we've accomplished this, we can take growth from the confluent area of the lawn of growth and use a loop and then put that in a tube to make a suspension in 70% ethanol. This suspension is made in special tubes that have metal cylinders in them to help homogenize that culture. So again, we said homogeneity is very important. So we'll vortex that culture and make sure it's homogeneous. We add water to those to make a final suspension and then rehomogenize that biomass by vortexing again. These suspensions are then spotted onto a 96-well graphite plate along with standards. This plate is then allowed to dry slowly so that the spots are just solid biomass and that the ethanol is allowed to evaporate. And during this drying time, you actually can go enter all your isolate information into the software and set up the run. So, while you're just sitting there having this go through the process of drying out, you can be productive and still do other things, such as entering all this information in. The isolate information can be set up then so that isolates from a particular facility or customer are kept separate from all the rest in the database. That was another key thing for us. We didn't want different facilities' isolates getting compared to each other. So you can easily set that up, setting them up as different species if you want, even for different facilities and different customers. That way you can avoid any kind of crossover when you do analysis later. We'll then set up the run in the software with all that isolate information and start the run. And then it only takes about an hour and a half for a full run of 30 isolates with 3 repetitions each. So it actually scans this pretty quickly and gets you data back pretty quickly as well. So then once all this data is generated by the machine, the computer will actually analyze that with hierarchical cluster analysis, or HCA. And then you can look at your data in various ways. Most commonly, people use a dendrogram or distance matrix that you can then export in a PNG format. There's other things such as heat maps as well. But here at Certified Group, we find these 2 formats for data analysis and comparison strains to probably be the easiest to look at and to understand. Especially when conveying this to customers, it's easier to kind of look at it in this regard so that they can see clustering of different isolates from different strains, and they can also see clustering of different species, even if you have different species here as well. So, the main thing here is to look at the clusters of isolates and determine relatedness. And this is really the main component of that analysis component is really being able to determine that relatedness and determine if isolates, are comparing to one another and if they're related to one another. So this will help you then determine things like if you have a harborage site in your facility where you're getting consistent sources of contamination, or if you're getting repeated contamination over time from some external source as well. So that's really the power of this in being able to determine where that source of that organism is in your process. So how does this help? Well, as I mentioned, once you have that information as to, if there's maybe a source within the facility that's causing this contamination or some sort of external source, you can then go on the hunt. That's the fun thing to do with this, is to then go and find the hot spots in your plant where that contamination is harbored and help eliminate those spots. That could be, pathogens, it could be spoilage organisms, a lot of different things. I get called out quite a bit to come help customers with these issues. They may say, hey, I've got contamination problems, and then maybe we've used some sort of subtyping technology like the Bruker to help narrow it down and say, hey, these are the places to look at. Well, then I'll go into their facility and try to dig into their equipment, dig into their drains and walls and all kinds of structural parts of the plant, and find out where this is coming from. And if we can actually identify it, then they can actually go ahead and fix it and clean it and rectify it and make sure that it doesn't come back. Some other uses for this are looking at whether we have relatedness of isolates when we have a major contamination event. So one other type of facility we work with is beef cattle slaughter facilities, and they have things called event days, and we'll actually talk about that here in the next few slides and give you a case study where that actually occurred. But these are massive contamination events, and we want to be able to see if these isolates are linked to each other sometimes. And so that can be very helpful using the Bruker-IR biotyper to be able to do that. Also, we want to maybe look at whether spoilage issues in final products are originating from a particular raw material, and we don't want to have to identify that organism either. Maybe we want to be able to just know, is the same organism there in multiple different lots coming from different raw materials, and you know, where did it come from in that process, and which raw material contributed it. So I've got some case studies in the next few slides where we'll dig into each of these different situations a little deeper and give you some real life perspectives on how we use the technology, the Bruker-IR biotyper, to accomplish some of the, I guess, challenges, if you will, that we were presented in each one of these case studies. All right, so here's case study number 1. So this one is actually, we'll just call it Facility A. They had multiple Listeria monocytogenes positives in a ready-to-eat sliced deli meat product and on food contact surfaces from that production line. One thing to note is that all positive product and product that had come into contact with the positive food contact surface had been destroyed. Because their product was considered adulterated, the facility went ahead and destroyed everything. So they still had these isolates to work with, but it wasn't going to implicate any product or their production facility. So they just wanted to figure out what was going on. And they had stopped production on the line until they could find a source. And so they wanted to determine if the Listeria monocytogenes that they found in their product was the same as any of the isolates that they found in their production environment, so that they could go about eliminating the source of this Listeria monocytogenes that was getting onto their product from this production line. All the samples at this point in time had already undergone cultural confirmation and were identified as Listeria monocytogenes. So the purified cultures that we had available from this were already identified and were already available, so we didn't have to do any sort of isolation or streaking. So we took these purified cultures and then streaked them heavily onto tryptic soy agar plates to create lawns, incubated those aerobically at 35 Celsius for 24 hours and obtained sufficient growth, and then went through the normal Bruker-IR biotyper protocol, where we took this growth and resuspended it in the Bruker-IR biotyper tubes, and then spotted those suspensions onto the Bruker-IR biotyper targets, allowed those to dry and analyzed them. And after analysis, we grouped all the isolates together into a single project and performed cluster analysis. So the output from the Bruker-IR biotyper was really nice because it allowed us to see that there were 2 major clusters, cluster 21 and cluster 13. Isolates in cluster 21 were A, B, and D. A was the product isolate, and B and D were environmental sponge samples from a slicer and the conveyor that was attached to that slicer. So we were able to quickly see that the slicer and the conveyor that was associated with it were related to the product isolate, and we're able to narrow it down to that region of the production line. This also showed that isolate C, which was from an environmental sponge sample of a utensil, was not related to the other 3 isolates. So that was also very helpful too with regard to the process of elimination. So based on the data from the IR biotyper, one was then able to determine that the slicer was the main source of the contamination of the ready-to-eat finished deli meat product. And although environmental sponge samples of utensils that were positive for Listeria monocytogenes were also thought to be a source of contamination, this data ruled it out. Thus, the client was actually able to focus all their efforts on taking the slicer out of commission, cleaning it really well, and they were able to ensure that it was ready to come back into service before the line was started up again. After this, there were no more product positives for Listeria monocytogenes. This was a big success story. Before using the IR biotyper, the options for analysis would have been PFGE or riboprinter analysis. and the PFGE runtime would have been 18 to 19 hours and at least 1 day to prepare. So the total analysis time would have been at least 2 days. In addition, those PFGE results could have been tied to regulatory databases. Riboprinter analysis would have taken at least another 8 hours to complete and was much longer than the time needed for IR Biotyper. So as we mentioned, this production line was down, so they needed to have results very quickly. So in this case, the IR Biotyper was able to take those isolates, quickly get similarity analysis comparisons done with them, and was able to help the customer make a solution to the issues that they were seeing with regard to contamination. So time is money in food production, and so this one was actually able to help you out very quickly with regard to time. The second study that was looked at here, the second case study, was with Facility B, we'll call them. They routinely test beef trim, which is meat destined to be used for ground beef, for E. coli 0157H7. And over the course of 3 days, they experienced 68 positive samples. So as you can imagine, this caused them to be in what we would call a high event period, or a very massive contamination event. Normally, E. coli 0157H7 positives are only about 0.3% of all the samples that are tested. So that is very low, and this was a very high percentage of positives. So Facility B knew that something went drastically wrong in their process, and they knew that they needed to troubleshoot it. So they wanted to actually know, though, if these isolates were linked, which would suggest a common source, before they went through this troubleshooting process. So just as in case study number 1, we were very fortunate to already have all of these isolates culturally confirmed positive, and they were positive for E. coli 0157H7. And then we took these purified cultures and went through the same process like we did with those Listeria monocytogenes isolates in case study 1 to prepare them for analysis with the Bruker IR Biotyper. When we did the analysis with the IR Biotyper, there were 6 distinct clusters observed. Cluster 412 was the largest, with 61 isolates that were in it. Cluster 408 only had 2 isolates, and then cluster 406, 370, 313, 354, and 305 all had 1 isolate apiece. So at the end of the day, these findings suggested that 91% of the isolates obtained during this high event period were highly related to one another, because they were all in cluster 412. And so based on this, it was hypothesized that a common piece of equipment, such as an inline fat analyzer, was contaminated, and that this was leading to multiple days of massive contamination events for this customer. So based on these findings, Facility B did an extensive cleaning of the facility, and they were able to resolve the issue that caused the widespread contamination. They had no more days of high-level positives after these corrective actions were made. And so in the past, the analysis of strains like this would have been done with PFGE. And that would have been much slower to complete given the sheer volume of isolates. I mean, this was a lot of isolates, 68 isolates that we would have had to run through PFGE. And only 12 isolates can be run at a time on a chef mapper that's used for PFGE. And each chef mapper run, as we saw in the previous case study, takes at least 18 to 19 hours, including at least 1 day of preparation before running. Thus, this could have taken us up to 8 days to get all of these preparations and runs completed. However, by using the IR Biotyper, we were able to run all 68 isolates within about 6 hours and have them ready for comparisons. So that was exponentially faster. And that throughput was very, very much appreciated by the customer, because it then allowed them to make their corrective actions sooner and get back up and running then very quickly thereafter. So the third case study we'll talk about is with Facility C, we'll call them. They had been experiencing ongoing issues with high aerobic plate counts in their blended cheese product. And these were causing the product to be out of specification to become spoiled over time. So not a pathogen issue in this case study. This one was actually a spoilage issue. So Facility C wanted to determine the source of these high aerobic plate counts by tracing backward from the finished product to individual ingredients. They didn't want to know the species of the organisms that were present, though. So they did not have this product on hold, and it was of high risk of triggering a recall if we found a pathogen. So the Bruker IR biotyper, with its lack of speciation, was an ideal situation for them here to be able to track whether this spoilage organism was coming from a particular raw material and then going through the process. So the customer then sent us samples of 9 different ingredients, 2 in-process samples, and a finished product package that all had a high microbial count. All these samples were then plated for aerobic plate count testing, and then we examined these plates and picked up to 7 isolates of different morphologies from each sample and streaked those to make lawn plates, which were then used to prepare suspensions for the Bruker IR biotyper. We then went through the standard protocol, spotted the suspensions onto the target plates, allowed us to dry, analyze them, and looked at the cluster analysis as a project again. 15 of the 22 clusters that we got out of here had only 1 isolate, so there were some isolates in here that just were not linked to anything else. 2 clusters actually showed that different raw materials had the same organism present. And this wasn't too surprising because we had several different cheeses that were raw materials that were coming into this blended cheese product. So the fact that we had 2 different organisms and 2 different raw materials, not too surprising. However, we had 4 clusters that had certain raw materials that had the same organism present in them, along with various work-in-process products. So there were several raw materials where the organisms were actually getting into the work-in-process samples. And then 1 cluster we had actually showed that the same organism was found in a raw material, an in-process product, and a finished product sample. So this was likely our spoilage organism that was causing the finished product spoilage problems that was coming all the way through this process. And so we were able to then tie it back to a certain raw material and saw that it was able to survive as it went through the in-process product and then went all the way to the finished product. So in conclusion, analysis with the IR biotyper was able to demonstrate that certain organisms were present in various raw materials, some of them that were present in various raw materials and in-process products, and then some organisms were present, and then in particular this one that we were just talking about, were present in in-process or raw materials, in-process products, and in finished products. And so through this analysis, Facility C was actually able to determine that the source of the spoilage organism that was observed in the finished product was a particular raw material. This spoilage organism was passing through that entire process, from the raw materials to work-in-process samples to finished products, and they were postulating that this was likely a spore former that survived heating because there was a lot of heating done in this process. And so that spore former was getting all the way to the end and then growing out and creating spoilage problems. Before the advent of the IR biotyper, this would not have been possible to show that these various organisms that we isolated from the samples were related to one another without having to do some sort of biochemical identification process. This could have actually identified the species if we did that. And so that was not something that the customer wanted to do here. So it was very nice that the IR biotyper was available to them to allow them to compare these organisms to one another without needing to biochemically identify them. And this was all simply done by just determining the similarity of the chemical compounds that were present and being expressed in their cell surface. And so this allowed one to determine that they were related to one another without actually needing to identify their species. And if one would have identified the species, one could then have identified a pathogenic species, and that could have caused all that product to be implicated in a recall, which the customer did not want. So this was a perfect solution for them to be able to figure out this challenge without getting themselves into any kind of regulatory problems by doing the analysis. So our last case study is with Facility D, which is an ice cream manufacturer that does routine monitoring for Listeria species in their processing environment. They also perform confirmation up to the point of a Listeria-like organism on a plate. And so they wanted to actually begin subtyping these Listeria-like organisms to be able to determine if they have one strain or if they have multiple strains. And they also wanted to be able to observe their contamination patterns. However, they could not know which Listeria species these isolates were in order to, again, avoid a recall situation. Because the product that they had produced was done while they had been collecting these isolates, and they didn't want to implicate a new product. So we actually had isolates from modified Oxford agar or Mox agar plates, which were typical Listeria-like organisms, but they had not been biochemically identified to the species level. They sent these isolates on plates, and these were frozen for banking at our facility. At these various intervals, isolates were streaked from these frozen cultures to tryptic soy agar plates in order to create awns, and then we went through the normal process of getting those prepared for the Bruker IR biotyper, as we mentioned in the previous case studies. So over time, Listeria-like organisms from this facility were grouped into 5 different clusters. These represented 5 different strains that were present in the facility. The largest cluster of these was cluster 184 that had 19 isolates in it, followed by cluster 182 with 8 isolates. And the presence of these 2 clusters demonstrated that there were 2 major resident strains that had taken up harborage in the facility over time. Cluster 164, cluster 146, each had 2 isolates, and cluster 167 only had 1 isolate. So these were all presumed to be some probably transient strains, more or less. Definitely not as persistent as those 2 strains that were represented by cluster 184 and cluster 182. So with this information, the customer was then able to plot all this subtyping information on top of their positive results that they were seeing with their routine monitoring, and were able to find some of the issues that they were having in contamination patterns that they were seeing over time. So Facility D actually took their positive point data from their environmental monitoring software, which they get through us, which I mentioned is the EMA program. They actually printed this off and then added the subtyping information on top of their positive spots on their maps. This then allowed them to be able to better visualize the distribution of each strain throughout the facility. And so over time, they kept doing this and kept adding these isolates to their database, and we're using this to determine whether a new positive result was due to a new strain being introduced into the facility, or if these were, or if a new positive was due to one of the 2 resident strains that were in their facility. In the past, this really could not have been done without identifying the species of this Listeria-like organism that we were actually analyzing. And so riboprinter analysis actually would have identified the species based on the pattern assignment. And so this was really ideal for this Facility D to continue tracking their strains without determining the species and implicating any products using the IR biotyper. So those were 4 different case studies of how we have used the technology in the past, but we also do see some future possibilities for how we will use the technology going forward. One of the things that our team will continue to do day in, day out is to use this on routine analysis of isolates that come in and need to be subtyped in order to determine if they are similar or not. I mean, that's what we're currently using it for right now. We're going to continue using it for that. However, over time, we would also like to do things such as developing a database of Salmonella serotypes and E. coli O groups to actually maybe potentially provide a rapid serotyping library for each of these isolates if one desires to rapidly determine the exact serotype grouping of the organism and do it pretty quickly. That would be nice because we see a lot of the similar cell surface structures between certain serotypes, and we would be able to group something in here without having to use a lot of anti-sera and doing a lot of extensive agglutination testing to be able to figure out what serotype it is. We also would be able to possibly do side-by-side studies with banks of isolates that have already been subjected to whole genome sequencing in order to determine the discriminatory power of the Bruker IR biotyper as compared to the gold standard microbial subtyping method of whole genome sequencing. We'd also like to understand that and how those 2 relate to each other. We also have thought about potentially using this technology in a research setting to see if different chemical structures on the exterior of the organism change over time, that being the same organism, and seeing if they change over time if they're subjected to different conditions such as stressors, chemicals, growth rate, etc. So that may actually alter some of that surface chemistry. And that would also be kind of neat to be able to know if that same sort of response is seen when those conditions are applied to different organisms or strains, and seeing if they have some sort of a shift in their chemical structure too. Again, a purely research-oriented type of situation there, but it might be useful in that regard as well. But again, like we said, the main reason for using the technology is to continue to do that routine analysis on those isolates and compare them to one another to see if they're similar or not. So again, I thank you for sitting through the presentation today. Hopefully we can answer some questions for you all after this, if you have any. And always please feel free to reach out to me. I'm very happy to always discuss this technology and answer any questions that anybody may have about it.